like to introduce Dr. Maryanne Platt. She's a postdoctoral researcher from Columbia University, and she'll present her findings from immune mechanisms and brain dysfunction in the mouse model of autoimmune encephalitis. Dr. Platt.
Hello, thank you for the wonderful introduction. For the past few years, I've been working on an animal model of post-infectious strep autoimmunity targeting the CNS, mostly looking at both the immune and the neurological dysfunction that occurs in this mouse model. So I'm going to start sort of broadly in the immune realm, and then talk about how this ties into autoimmunity, and then talk in the more nitty-gritty data side of things, what I've actually worked on, and then sort of broaden it back out, getting back to what other people have done, and sort of is this suggestive of how other people, or of how other autoimmune encephalitis conditions might work. So this is pretty basic immunology, but bacterial infections promote expansion of different T cell populations depending on the pathogen to control that bacterial infection. So group A strep, as you've seen a million times here in this lovely blood agar plate in, this nice little pearl necklace, but it gets processed by these antigen-presenting cells and chopped up into smaller pieces that can be shown to T cells that are, as we call them, naive. They haven't ever seen an antigen before. And these antigens are little bits of bacteria that are unique to the bacteria and tell the T cell what to look for and what to kill. So these T cells expand in response to being stimulated by this antigen-presenting cell. And the classical directionality is that these T cells then inform B cells, and B cells produce antibodies against these bacterial antigens. And more recently, some work has shown that B cells also circle back and talk back to the T cell populations to sort of sculpt that immune response, which has been shown to proceed very nicely and results in T cell activation and T cell memory, so persistence of T cells that stay in the body for a long time and help the body remember that it's previously seen that same bacteria. And when we talk about these different immune populations, they all develop from the same naive precursors that live in lymph tissue, like your thymus and your lymph nodes. But depending on how these T cells develop and what subtype they eventually differentiate into, whether it be Th17, Th1, Th2, Th22, Tregs, follicular T cells, they secrete different cytokines, which are inflammatory molecules. These cytokines both talk to other immune cells and other immune populations, but they can also have sort of off-target effects on other tissues in the body, which I'll come back to later. But strep infections really promote expansion of Th17 cells, which release IL-17A. And I know you've heard this a zillion times today already, but it's very important, and I'm going to be talking about IL-17 a lot. So autoimmune encephalitis occurs when these antibodies recognize brain antigens, the most common of which is the NMDA receptor. Over a dozen targets have been identified, and symptoms are very varied and diverse, even within the same subclass of autoimmune encephalitis. But seizures, memory problems, a long range of symptoms, and you can see that these antibodies, when they're allowed to react with neurons in a dish, react very strongly and tightly bind onto their neuronal targets that are on the neuron surface. So the most well-described autoimmune encephalitis targets the NMDA receptor, which is heavily implicated in memory formation. So as a result, these patients typically don't remember any of their disease course. And I'm not a clinician, but this is how I sell my talk to non-clinical audiences when I have to sell it. So these triggers are very rarely identified, actually. Only in about half of cases are triggers for these autoimmune encephalitides ever identified. And it sort of breaks down into two different classes of triggers. The first of which, as you can appreciate, is tumors. So ovarian teratoma, which is a tumor on the ovary, is very strongly linked to NMDA receptor encephalitis. And indeed, in the first cohort of NMDA receptor encephalitis patients, that was almost an inclusion criteria, was presence of an ovarian teratoma that expressed the NMDA receptor on those tumor cells, in the ovary. But thymoma, so a tumor in the thymus, can also be linked to several different autoimmune encephalitis conditions. And the other class of trigger involves infection, so bacterial and viral infections have both been implicated in development of autoantibodies against anti-RO antibodies and the dopamine receptor, obviously, and then in NMDA as well. But what do these two trigger classes have in common, right? They're both present in the outside, in the periphery, or i.e. not the brain, right? So the immune system is able to see things that are present in the rest of the non-brain body. So these triggers all allow the immune system to sort of see these triggers and react with them before they go and attack the brain. So the important question that brought our lab into this game was, how do these autoantibodies that are heavily implicated in disease pathogenesis access their CNS targets? For example, the NMDA receptor encephalitis antibodies are present in about 10% of the population overall, but the incidence of NMDA receptor encephalitis is only about 1 in 100,000. So there's got to be some way to make these antibodies pathogenic, and that is that they can leak out of the bloodstream where they're present. So normally, these antibodies are excluded from the brain by the presence of the blood-brain barrier. So the blood-brain barrier is the focus of Dritan's lab, but it's a very important gatekeeper between the blood and the central nervous system, and it can be visualized if you inject a dye tracer. So in this case, we're injecting a blue dye called Evans blue into the circulation of the mouse. And then if you take out various organs, you can see that the blue tracer is taken up by the kidney and the liver, where it leaks out of those capillaries and stains the tissue. But in the brain, this dye is excluded from the tissue by the presence of the blood-brain barrier in these brain capillaries. And the BBB is very important for maintaining brain homeostasis. The brain needs certain concentrations of glucose and oxygen. It's a very needy organ. It also needs very particular concentrations of ions, calcium, potassium, chloride, all those good things that are important for neuronal signaling. And the BBB also limits CNS entry of pathogens, so it keeps infection out of your brain, as well as toxins and drugs, much to the chagrin of every pharmaceutical company, and immune cells. So the brain is very uniquely not very surveilled by the immune system, contrary to the degree that the rest of your body is. So, the question that we're sort of arriving at is, how do these brain-targeting autoantibodies access the CNS, and how does the immune system sort of begin to infiltrate the brain in autoimmune encephalitis caused by infection? So T cells are present in the brains of various autoimmune conditions that target the CNS. So the most classical presentation of a CNS autoimmune disease is multiple sclerosis. And in this disease, T cells get activated targeting myelin, so targeting brain proteins present on neurons in the periphery, in a peripheral lymph node, and then the Th1 and the Th17 cells that circulate secrete cytokines, particularly the Th17 cells are very involved in this process, but the cytokines that Th17 cells secrete act directly on proteins that seal together the blood-brain barrier, and they break the blood-brain barrier themselves, allowing both those Th17 and other T cells, so Th1, to leak into the brain and begin to secrete other inflammatory molecules and enact this myelin destruction that is classic MS presentation. So do we see T cells in autoimmune encephalitis brains that are not MS brains? And the answer is yes, we do. And these are two post-mortem brains from two different autoimmune encephalitis, non-PANDAS, but targeting two different brain antigens. So here in this top image, you can see T cells. This is CD3, which is a marker for all T cells, collecting perivascularly, so sort of surrounding this blood vessel in the middle. They're sort of collected in this space between the actual blood vessel and the membrane that limits, that's between the vessel and the brain, as well as in the brain tissue, frankly, itself. And then in this other case, they're in the brain parenchyma, infiltrating in with neurons and glia. So given that we're seeing this T cell infiltration into the brain in autoimmune encephalitis human cases, we want to figure out a way to bring this back into the lab, to study it in a mouse model where I can actually manipulate the mouse, look at what's going on during the course of disease, look at before I know the disease is going to set on, and then look at things down the line and how the disease develops. So we chose a post-infectious autoimmune model where we know that group A Streptococcus is heavily implicated in autoimmunity. You've seen this slide in some form or another in several different ways today, but autoimmune disease can develop targeting, the heart, the kidney, the skin, if you're looking at scarlet fever, and then targeting the brain in Sydenham's chorea and in PANDAS. So we wanted to choose something that we know, or we wanted to choose this infection that we know is rheumatogenic, we know causes autoimmunity. So we adapted this protocol from Pat Cleary's lab, where we're taking young mice, so these are just after they've been weaned from their mother. At that point, we can separate them out so that we're not infecting our control group in the same cage. And we give them weekly inoculations of group A strep or PBS, which is just saline, which is the quote-unquote our vehicle control. It's what we put the strep in. And we do that once a week for 5 weeks. We just use a little dropper to drop the strep right into their nostril, and they sort of inhale it as they're breathing. They don't love it, it's not their favorite thing, but it doesn't hurt them in any way. And then at the end of the 5-week protocol, we can examine what's going on in the brain and look at a variety of different immune outcomes as well as the neurological outcomes. So what we see in the site of infection, so in the nose, is in this top image you can appreciate we're looking at, this is the NALT, this is the nasal-associated lymphoid tissue, it's a mouse analog of the adenoids and tonsils. And then in the olfactory epitheliums, this is these lacy, they're called turbinates, where the neurons that sense smell reside, and they're heavily patrolled with immune cells, but strep collects here in green in the mucus, this is like mouse snot, and then T cells collect right around the strep in the olfactory epithelium. And we sort of developed this model where we realized that the T cells that are in the olfactory epithelium here in the nose are a mixed population of Th1 and Th17. This was work that Depletion and Pat Cleary did a couple years ago. So we're looking at this population that's a mix of Th1 and Th17 in the nose. And then we wanted to take that into the brain, right? Because we're a brain lab, that's the most important organ, obviously. So we first looked for the presence of T cells in the brain. We didn't know what to expect. And what we found was, first look over here on the left in this image, this is the anterior portion of the brain called the olfactory bulb. And in our control mouse, you can see here these nice spheres of green. These are where the sensory neurons that sense smell, these olfactory neurons, that's where they land. And you can see it in the mouse that's been infected with GAS, with group A strep. You see these red T cells stained for CD4. So these are helper T cells sort of along these axon tracts and then also landing within this brain region here. And if we quantify the number of T cells that sort of exist along the rostrocaudal axis of the brain, so from the nose side back to the posterior part of the brain, this is a side view of the mouse brain. So up here, this little lobe is the olfactory bulb, and this is the cerebellum, sort of like right below the ears. And this red line through the olfactory bulb corresponds to this dashed red line. This blue line corresponds to this blue line, so you're kind of oriented. Most of the T cells that we see this massive wave of infiltration in the anterior portion of the brain, which is right near the nose. So like this A is like the thin, spongy part of the bone of the skull that separates the brain from the nose. So these T cells are really coming from the nose right into the brain. They're landing here in the olfactory bulb and then kind of migrating more posteriorly into these brain regions where there are still significantly high numbers of T cells compared to uninfected controls, which are here down at essentially 0. So we're seeing this massive wave of CD4 T cells in the brain. This is all old data. This has been published for years. And I'm going to just summarize this with a model. I don't want to go through all the primary data. It's available online at the Journal of Clinical Investigation. But these T cells that make it from the nose, they migrate into the brain, so they're crawling along these olfactory axons where they're sort of landing in the olfactory bulb and they're secreting these inflammatory cytokines, which are these blue and red dots. And these cytokines act on the cells of the blood-brain barrier, so on the blood vessel walls, where they disrupt the cell-cell contacts that kind of hold the adjacent cells together. in the blood vessels, and that allows both blood-borne antibodies and immune cells to leak out into the brain. So you get serum antibody deposition in the brain, and then the further infiltration of more immune cells causes the native immune population of the brain, the microglia, to sort of react and round up, and they start to clean up all the debris that's being deposited there. And then as we've sort of discovered more recently, we see a decreased activity of the olfactory sensory pathway. So neurons that sense smell and tell the rest of the brain about the sense of smell are actually impaired in doing their job and impaired in their signaling. For the rest of the talk, I'm going to be talking about unpublished data, so I'm going to ask, again, not to share things widely. I'm actively working on revision experiments for this publication, so just, if you're going to take pictures, but please don't share them, at least until the paper's published, at which time, yes, share them far and wide, as Dr. O'Connor said. So Dupont and Erica and Pat Cleary's lab showed that the strep-specific T cells in the brain and in the nose were mainly this Th17 variety. And when I joined the lab, Dritan pitched me this project and said, hey, do you want to look at what Th17 cells are doing? And I said, yes, very much so. That sounds very interesting. So I looked into how I could do that, and it turns out that T cells develop into their sort of final form through the expression of different transcription factors, and these are proteins that turn on or turn off genes that control the fate of that final form. So the gene that sort of permits Th17 fate is called ROR gamma T. The name doesn't really matter. It's an orphan protein, but what a clever lab has done is created a mouse line where we can manipulate the expression of ROR gamma T and either visualize Th17 cells and where they are in the brain, in my case, or we can eliminate ROR gamma T and completely eliminate the presence of Th17 in the body. So in this mouse line here, which is called the ROR GFP line, the wild-type mouse has 2 copies of the ROR gamma T gene, like most genes, in what I'm going to call ROR hets. They have 1 copy of ROR, so Th17 cells can still differentiate, they can still develop normally, but it has 1 copy of GFP, which stands for green fluorescent protein, and that's exactly what it does. It fluoresces green, so in all of these normally developed Th17 cells, they're labeled with green. So I can track them and I can see where they are in the brain. And then in ROR gamma T knockouts, both copies of this ROR gene are replaced with GFP, so Th17 cells can't differentiate, they can't develop, and we've effectively eliminated them. And what we can actually do is isolate the contribution of Th17 to disease processes if we look at what disease looks like in these knockouts. So anything that's not there, we know is due to the presence of Th17. So I started by infecting ROR hets up here on the top and ROR knockouts down here on the bottom. And I looked for, first in ROR hets up at the top here, there are some green cells and some red and green cells, indicating that we have both Th1 and Th17 in the brain, both in the olfactory bulb, in that anterior part of the brain, and in the choroid plexus, which is a little bit farther back, and it's another important epithelial barrier in the brain. In ROR knockouts, we only see red cells, so we don't see any green Th17 cells. We really only see strictly, well, we don't know they're Th1 yet, but they are. And if we count the total number of T cells, this is a similar graph to what I showed before, but in the anterior regions of the brain, in the ROR hets, which are normal immunologically, that we recapitulate this robust infiltration of T cells into the anterior parts of the brain. And in the ROR knockouts, we see about a 50% reduction in the total number of T cells that are present in the brain after recurrent strep infections. But it's unclear from this data what the identity of these remaining cells are. So what I did was I typed them based on what cytokines they express. I looked at what cytokines of IL-17, IFN gamma, and then 1/3 portion of T cells called Tregs, which are sort of anti-inflammatory. I saw an increase in Th1 cells, so interferon gamma producing, and then a decrease in all of the populations that express Th17. So we really eliminate this Th17 fraction. And what you get is sort of a picture like this, where we had, a lot of cells expressing both IFN gamma and IL-17, some Th1, some Th17, and some Tregs. And then in the ROR knockouts, we see almost exclusively Th1 cells that only express interferon gamma, to the detriment of all these other T cell subtypes. And then in the NALT, the picture is much the same. So in the nose, we really have exactly the same phenotype, where we're increasing the Th1 population and decreasing both Th17 populations that we're seeing in the nose, suggesting that these cells that are landing in the brain are originating in the nose, and they're not coming from the circulation or somewhere else. So my next move was to look at if Th17 cells are controlling blood-brain barrier permeability to either blood-borne solutes, blood-borne antibodies, or other solutes. And just in the interest of time, I'm going to just only talk about antibodies. So this is sort of a cross-section of a blood vessel. This is just like 2 cells hanging out next to each other. And then antibodies that are present in the blood are trying to get through over to the brain side. They can either go through the zipper contacts that seal together those cell junctions, or they can go through the cells being transported by like cargo ships that sort of traffic cargo through the endothelial cells into the brain. So those are really the 2 routes they could be taking. I'm not going to differentiate between those 2 routes, either between the 2 blood vessel cells or through the cells. We've done a little bit of work on that, but it's a little complicated for right now. So if we map areas, so what I've done here is recurrently infect with strep in either ROR hets with Th17 cells or ROR knockouts without Th17 cells, and then looked for deposits of IgG in their brain. And all the data I'm going to show is in the olfactory bulb, where we see the most T cells and the most prominent pathology. And if we map areas where there's IgG deposition, we see that the area of IgG deposition is relatively unchanged between the hets and the knockouts. So there's still a wide area where the blood-brain barrier is leaking. But what I hope you can appreciate in this slide is that in our control mouse, so the blood vessels here are in red, and you can see they're kind of like spiderwebby. And then IgG is in green. So our control mice have no IgG in their brain. Great, that's normal. Our strep-infected ROR hets have kind of a lot of IgG that's being sort of stained on these neurons in the brain in 2 different brain regions. But when we infect gas ROR knockouts, we pretty much eliminate deposition of IgG. It's still there, but it's much less intense. So either the IgG that's being trafficked in is being pumped right back out again, or there's a much smaller leak in the blood-brain barrier here to allow fewer IgG molecules into the brain. And when we quantify that, we see that it's very significant. We have, this increase in intensity of IgG deposition, and then almost a rescue of IgG intensity in the ROR knockouts without Th17 cells. So it seems that the Th17 are almost controlling how much antibody is allowed into the brain. So this is the fun part. This is the very cute part. So I saw that all this pathology was happening. I also tested microglial activation and barrier permeability to a couple other molecules. But the most prominent pathology that we were seeing was all in the olfactory bulb, where and information for the sense of smell is processed. So I wanted to test and see if the neurons that live in this area were still able to function after recurrent strep infections. So what I did was, I can ask a mouse if it can smell, but it's not going to say anything. So I have to ask it a different way. And what I do is this test called the habituation-dishabituation test. It's a long, fancy name that I hate saying. But essentially what I do is I give the mouse a Q-tip in its cage, and the Q-tip is either unscented or impregnated with a little bit of odor, and the mouse will sniff, right? The mouse is naturally curious, so it's going to sniff at a new thing that it's never seen before in its cage, or it's going to sniff at a new odor never smelled before. So the time that it spends sniffing the odor is indicative of interest, and interest is indicative of brain function. You have to be able to smell something to be interested in it. So the mouse is presented 3 times in succession with the same odor before a new odor is introduced. So, the first time you smell something, it's like really novel. You want to smell it a lot if you're a mouse. So the first time the mouse is presented with just a water-scented Q-tip, which doesn't smell like anything, it just smells like Q-tip, it sniffs at it a lot. It's very curious. But then the second time it sees it, it's like, well, I've seen this before. It's still there. Cool. And the third time it sees that same odor, it doesn't care anymore. It's old news. But then, I change the odor. I give it an odor. I give it an almond scent, and this is just almond extract I bought at Whole Foods. So over the 3 successive trials of the same odor, it gets disinterested. It habituates to that odor. And then at the initial presentation of a new odor, it dishabituates. It gets more interested in that odor, in the new odor that it's never smelled before. So by comparing amount of time spent sniffing each individual odor trial, we can ascertain if the mouse can smell. So if it can smell, it will have a bump here at the first presentation of odor. And if it can differentiate between different smells, in which case it'll have a bump at this second odor when I introduce a different odor from the first one. So I did this in my inoculated mice, and what I saw was that control mice smelled very well. All of the mice were interested in the Q-tip, which was great. They could all see it. They were all interested in a thing that was new to them. So it's not, for lack of interest, and it's not because these mice are acutely ill. But interestingly, strep-infected mice here in black and in red can barely detect any odor, and they certainly can't differentiate between new odors, whereas the PBS mice that were not infected can, indicating that these mice are somewhat anosmic and that their sense of olfaction is impaired. And I was like, oh my God, this is great. More neuroscience. What I did was I wanted to listen to the neurons that are actually firing and actually sensing that odor in the brain. And to do that, you stick a piece of metal in their brain. And the metal conducts the electrical impulses that signal between neurons, and neurons fire. They send electrochemical impulses between adjacent neurons when they're conveying information. So by lowering this probe into the right layer of the brain, and this is the mitral and tufted cells, so this is like 2 layers in from the nose, and this is the layer of cells that projects back to the part of the brain that like assigns emotional valence to things. So when you smell like roses or whatever and you think of your grandmother, these are the neurons before those neurons. So I took a whole bunch of mice and this was all performed in collaboration with Kevin Franks' lab at Duke University, where they have a whole neuroscience setup to record from mice that do olfactory tasks. And what I did was I lowered this metal probe into the mouse's brain, into the olfactory bulb, and you can listen from these circles are all different electrode sites. And you can listen to the neurons that are immediately adjacent to these electrode sites, and then hear from this, small, but I'd get like 15 to 20 neurons that I was listening to at the same time, while the mouse is being exposed to a variety of different odorants. It's like an awake mouse that's head fixed with this cone of odor in front of its face. So the data that I'd get would be a bunch of raster diagrams like this, and so each row here is one cell that I've decoded the data for. And at baseline, so before this black line, this is before the onset of odor, all of these cells sort of have a baseline firing rate where they're just happily going along, and they can either modulate that rate up or modulate that rate down. And at the onset of odor, you really get this robust increase in spikes. So this green is odorant. You get this robust spike in activity, and if you sum all these cells up, you get this nice odor-induced spike. So if I look at concentration different odorants. In control mice, you see this nice odor-induced spike in both ROR het and ROR knockout mice that are healthy. And then in strep-inoculated ROR het mice, we pretty much have ablated their sense of smell, so they cannot smell anything. Whereas ROR knockout mice have some ability to smell, that we've preserved some of their olfaction, indicating that Th17 cells might be responsible for acting on these neurons and really impairing their function in some way. And this is also affecting the ability to recruit more cells if you increase the concentration of odorant. So we sort of arrived at this model that's, imminently published, hopefully, where a pure Th1 population is recruited to the olfactory epithelium in ROR knockouts in comparison to the mixed Th1, Th17 population. And these interferon gamma releasing Th1 cells are much less efficient at breaking the blood-brain barrier, resulting in, much happier microglia that are just content to rest and not have to phagocytose as much. And then that preserves the activity of the mitral cells that are signaling odorant information back to the rest of the brain. So in thinking about, all of this, activity of different T cell subsets, and how pathogens might be influencing brain function, Dritan and I have been bouncing back and forth, thinking about what other people have done as well. And this is old data as well. This is from the Dialpon paper. T cell presence in the brain is a unique feature of strep infections. So if we look at mice that have been infected with strep, this is a similar T cell infiltration graph. We see this nice population of olfactory bulb T cells and elevated numbers of T cells throughout the brain. But that contrasts with mice infected with either Salmonella or Listeria monocytogenes, which don't induce nearly as robust a population of T cells, indicating that maybe strep has some sort of like brain tropism. The T cells that it induces certainly do. Throughout this process, the brain is not infected with strep, but that also got us thinking about, what about these other, tumor-induced encephalitides, like they're not getting infected with strep. Strep isn't the causative agent there. And excitingly, a paper from Gary Westbrook's lab just came out talking about NMDA receptor encephalitis in a mouse model, where they inject these mice either with the NMDA receptor sort of suspended in these like fatty spheres, or in empty they're control mice that have empty fatty spheres injected in them. And then they look at immune infiltration in the brain, and what they see in control mice is really no T cells, but in mice that were injected with these proteoliposomes, so the lipid droplets with the NMDA receptor, they see collections of T cells in regions of the brain that express NMDA receptor very highly. So this is the hippocampus, and it's critical for memory formation. It's chock full of the NMDA receptor. That's like what you stain if you want to see the NMDA receptor. So this was very exciting, and they quantified the number of T cells present in these brains in various brain regions, and they're kind of all over, but all of these structures express very high amounts of the NMDA receptor. And we also see elevated T cells and B cells in these NMDA receptor injected animals. So, immune infiltration might be sort of a common thing behind all encephalitides. That's sort of where our thinking is going. But in the future, we have a couple of avenues that we're still looking to investigate in the lab, both on the basic science side of things, and then also on like the clinical end. So sort of this B-cell, T-cell interaction hasn't really been studied in our lab, in our model, or in other models. And I personally would also like to really dig into the microglial question before I leave the lab. And then Dritan and sort of the human arm of Team PANDAS in the lab has been trying to test for Th17 cytokine signatures in sera and cerebrospinal fluid in PANDAS patient samples. We also want to develop novel imaging techniques and strategies so we don't have to rely on things that aren't necessarily specific for PANS and PANDAS, but this would allow us to visualize changes in barrier function and potentially develop new immunotherapies that might target IL-17 or Th17 besides just steroids that hit it with a hammer. So Thank you to my lab. Thank you to all of you. Thank you to my funding. This is the current lab now. This was at our biggest when we had like 22 people in the lab. It was ridiculous 2 years ago. Thank you. That's all.